Dear students, Assalamu alaikum to all of you. This is class 11th, Zoology, and I am Zuhuram Hamdani teaching you the topic for our discussion today is enzymes. Enzymes are the biological catalysts which are water soluble in nature, colloidal in nature, organic in nature, and macrobiomolecules, wholly or partially proteinaceous in nature. They are biological in origin, produced in micro amounts, and capable of catalyzing specific biochemical reactions under specific conditions of temperature and pressure. Enzymes are those biomacromolecules which in the functional state contain two parts. The first part is the apoenzyme. This part is proteinaceous in nature. The second part is the coenzyme. This part is non-proteinaceous in nature. When the apoenzyme and the coenzymes combine, they form a functional enzyme that is called as holoenzyme. This holoenzyme is functional in nature, which performs biological catalysts in biochemical reactions. The coenzyme can be, for example, in the physiology of respiration, nicotinamide adenine dinucleotide or nicotinamide adenine dinucleotide phosphate. These are also coenzymes which are non-proteinous in nature that make apoenzymes functional to make a complex holoenzyme. The enzyme action reveals that there are a large number of mechanisms by which the enzymes work. The most important mechanism, the model explains how do the enzymes work, that is log and key mechanism. According to this mechanism, just like a particular log can be opened by a particular key, so a particular biochemical reaction can be catalyzed only and only by a specific enzyme. That enzyme acts as a key for the catalysis. According to this mechanism, a particular enzyme fits into the molecule of a substrate in such a manner that there is an intimate association by their specific areas. These specific areas are called as reactive sites. These reactive sites bind. These reactive sites are present in the substrates that bind to the enzymes. These reactive sites are very specifically designed. Only specific substrate molecules with proper geometric shape fit correctly. They form a complex. That complex is called as enzyme-substrate complex. Enzyme-substrate complex. This enzyme-substrate complex, as per the kinetics of the biochemical reactions, reveals that this is a transient phenomenon. Transient phenomenon means that this enzyme-substrate complex is short-lived. Later on, it breaks down to form the enzyme again that can be reused in that very particular chemical reaction, and the product is formed from the substrate. That product is yielded in the chemical mechanism of the cells. Active sites contain special groups having amine group or carboxylic groups for establishing contact with the substrate molecules. Because particular law can be opened by a particular key, and particular enzyme can act on a particular substrate due to their specificity, the name for the mechanism has been given by lock and key mechanism. This name has been proposed by the scientist Fischer in 1899. I think this is sufficient for today. Thank you.